Thank you for joining IMG webinar series. The second part of it, my name is Natalia Ivanova, and today with me presenting the IMG Similarity searches are other researchers and software developers from the JGI, which is Joint Genome Institute, and these are Rekha Seshadri, Tian Chu, Amy Chen, Neha Varghese, and a few other people. A few words about how we are going to conduct this webinar. First of all, it will be a live IMG demo, which means that I will be trying to run some of the searches and queries live real time. And because of that, depending on the load of the system, the performance may be somewhat varied, and if so, which means that if we run a little bit over time, we scheduled it for 1 hour, but we may run a little bit over time. So we ask you to be patient with us and to excuse the performance of the network. So the second comment that I would like to make is that this webinar is being recorded. And the recording will be posted on the JGI YouTube channel together with the transcript of our question and answer session. So all of your mics are muted and you cannot unmute them, but you are most welcome to ask questions using the chat that you will see in the Zoom chat window. We will try to answer as many of these questions as we can live during this webinar. If there are too many questions for us to answer, we will still answer all of them and post them in the chat transcript. So you are encouraged not only to watch the webinar itself, but also browse this chat transcript, the Q&A. Also, even if you received an answer live during the webinar, we still encourage you to look at this chat transcript, because as we are going through it, we may come up with a better, more complete, or more precise answer. So that's kind of the format of the webinar. So the second part is that it will include, first of all, I will go through some introductory slides, and then I will go with the live IMG demo of IMG Similarity searches. In between these parts, I will stop and I will ask my colleagues if there are any questions that are asked by many participants. If there are such questions, we will answer them live on air. If there is nothing that is of common interest, we will proceed with the rest of the webinar. Now let's begin. So the very first slide is we have to say a few words about who we are. We represent the DOE Joint Genome Institute, which is a DOE user facility. DOE user facilities enable large-scale science to advance the missions of the Department of Energy, such as clean energy, bioremediation, and addressing various environmental challenges. So DOE Joint Genome Institute has been funded by DOE Office of Science since 1998, when it became part of the Human Genome Project. And we are conducting omics research, meaning that we generate sequence data for genomes, metagenomes, transcriptomes, metatranscriptomes, and so on. And we also have some metabolomics capabilities. As of now, we have about 280 staff. We are located in Berkeley, California, and if you are interested in what JGI is and what it does, we encourage you to go to our website. And let me go to the next slide. So this is a brief overview of JGI's contribution to the sequencing of plant and microbial genomes, which is our focus right now. So we are a major contributor in all of these areas. JGI is responsible for generating more than 20% of all plant genome sequences, almost 40% of fungal genome sequences, and more than 40% of archaeal and bacterial genomes, as well as environmental metagenomes. So all of the JGI is not only generating this data, it is also providing this data for users for analysis, download, and other capabilities. And you can find the plant and fungal genomes in two JGI portals called Phytosome and Microcosm, respectively. while the bacterial and archaeal genomes and metagenomes are found in the IMG EMER system that we will be talking about today. So the next slide is about IMG. And today I will be using for this presentation the secure system, or IMG EMER. IMG exists in several flavors. The completely public open IMG EM, although all of the data there is public, it still lacks some of the capabilities that are available in this system, which is called EMER, which requires login, which requires an account and login. IMG also has two portals that are specific for certain types of data, such as IMG ABC, which is the system with biosynthetic gene cluster information, and IMG VR, which includes the information about viruses. And how you can figure out that you are in IMG EMER is you will have in the upper right corner of your screen, you would have your username and logout link. In addition, you will have this option of workspace in the top IMG menu. And this workspace means that you can submit your data, annotate data, save results, access other workspace functionality, as well as submit computation jobs. And this is the important part that we will need for the demo that we are doing today. Now, if you try to get an IMG account, or if you got an IMG account, but you have difficulty logging in, then we ask you to go to this help link in the very corner of IMG main menu. If you click on it, there will be a contact us option. If you click on this link, you can submit a ticket so that we can address your problem. Now let's talk about what kind of similarity searches IMG provides. So these slides show the summary, kind of very high level summary of the similarity searches available on IMG. And it tells you that IMG is enabling two types of searches, BLAST and LAST. And these are the searches, pairwise sequence similarity searches. What IMG does not do for you, it does not allow you these profile-based searches, such as Psi, BLAST, HMM search, and CM search. Now let's discuss the main parameters of the BLAST and LAST searches available on IMG. First of all, the query sequence. You can use the query sequence that comes from IMG, or you can input your sequence of your own interest. So the sequence type can be either nucleotide or protein. And the databases against which this search will be performed, it can be either all IMG genomes, or it could be individual data sets, individual genomes, individual metagenomes. In addition, IMG has a collection of specialized databases, such as, for instance, this 16S ribosomal RNA. We also have a specialized database of viruses and CRISPR spacers. Lastly, let's talk about the search mode. There are many searches that you can conduct on IMG on the fly, meaning that while you are waiting for it and looking at the browser. In addition, IMG has this mode of requesting a computation, meaning that you submit the search and then it runs on the background, and when you get the results, when the results are ready, you get an email notification. Again, this option is available only to this request for computation option is available only in IMG EMER, only to users who have accounts and who have logged into IMG. So I'm not going to talk about the details of what is the BLAST and LAST algorithms. And here are two links if you are interested, if you are novice to this area. Here are the slides that describe graphically how BLAST algorithm works. And here is the link to the LAST website, which describes LAST algorithm, parameters, and other options. So although I'm not going to talk about details of BLAST and LAST algorithm, I'm going to stop briefly on the alignment scoring in BLAST and LAST. And this is important because it is somewhat a deviation from what people are usually thinking about how the alignment scoring should work. So both of them, BLAST and LAST, are conducting pairwise sequence alignment. And as a result, you will get something like this screenshot in the window, where you have on top your query sequence, on the bottom you have the subject sequence, the one which is similar to your query, and then in between you have the matches and mismatches. If there is an exact match between the two letters, like for instance this aspartate, then it is the letter. If there is a similar amino acid, such as aspartate and asparagine, then there is a plus, and if there is a mismatch, like between the cysteine and tyrosine, then there is an empty space. So this is the typical representation of alignment of this pairwise sequence alignment, a screenshot from BLAST. And after both of them conducted this pairwise sequence alignment, after that they go through the return the kind of the summary metrics of how good these alignments are. And there are three of them. One of them is this bit score, which is the sum of scores from the scoring metrics. By default, it is Blossom 62. Essentially, what it means is that the algorithm does very, very simple arithmetic. It goes through this matrix, the scoring matrix, and says, I found a match between cysteine residues. I will add 9. The score will be plus 9. Or I found a mismatch between cysteine and tyrosine. The score will be minus 5. So I will subtract 5 from this cumulative bit score. So that's the bit score. It is not equal. It is proportional to the sum of scores because there is some normalization based on the protein alignment statistics and so on. The second parameter that both of them report is this E-value, which is proportional to actually the inverse bit score and database length. And the last one is this percent identity, which is a very, very simple parameter. It is essentially the number of identical residues that you found in this alignment divided by the alignment length. Now the question is, today we will be looking we will be searching, we will be comparing full-length sequences from isolate genomes to fragmented metagenomics data. And metagenomics data is always fragmented because the average context size in the metagenomes is less than 1,000 nucleotides. So by default, you will have a lot of protein fragments. So the question is, out of these three measures of alignment quality, which one is most suitable for analyzing this fragmented metagenomics data? Again, our goal in this case is we are trying to find the sequences that are more closely related, that have less evolutionary distance and so on. And if you have ever taken any bioinformatics courses, you would probably assume that it is E-value or at least bit score, because both of these are statistical measures of the sequence similarity. Now, you may be surprised to find that it is not actually the case for comparing sequences to fragmented metagenomics data. Let's look at these two alignments. So the alignment on the left shows relatively distant sequences. As you can see, there aren't too many identities and there are a lot of spaces showing that there is a serious mismatch between the amino acids. On the right, there are two identical sequences. Now, what would you think the bit score be for the alignment on the left and alignment on the right? If you guessed that the bit score for the sequences on the left is lower than for the sequences on the right, you guessed wrong. Because although percent identity of the sequences on the left is only 55%, bit score is 47. While the sequences on the right with 100% identity, the bit score is only 38. And that's because you have to remember that bit score is a cumulative measure. It depends on the alignment length. The longer the alignment, the higher the bit score. So even though in many cases you would use bit score and E-values over percent identity, specifically for analyzing fragmented metagenomics data, percent identity is the preferred score to compare alignments and to compare similarities, to find the sequences that are more closely related. So that's the summary, right? So for comparison of metagenomic sequences, percent identity is preferred. So this is a brief overview of the sequence similarity options in the UI. And this is mainly for you kind of as a memory card when you are looking later on. You can see that the sequence similarity searches can be found in many pages, in many IMG pages. in gene details pages specifically, and we have different options depending on whether it is RNA or protein gene details page. Then there is a menu option called Find Genes, BLAST with 5 flavors. And lastly, there is this option of workspace with genome sets with BLAST tab, where you can submit a computation job which will run on the background and return the results by email. The topic of today's demo will be looking for Bradyrhizobium genomes in the environment. So what is Bradyrhizobium? Bradyrhizobium is a genus of root nodulating bacteria. It is a symbiont of legumes which fixes nitrogen to supply the plant host. And it is a remarkable biological system because there is a lot of interaction between the host plant and the bacterium, where the plant extends sort of a welcome note by secreting flavonoid compounds. And when these compounds are recognized by the bacterium that there is a suitable host in the environment, the bacterium in turn, the bacterium will secrete the its own signal called nod factor. And when the plant recognizes the bacterium as a friendly bacterium, it will turn off its immune response to bacteria and instead form this organ called nodule, where the bacteria will propagate and the plant will create the ideal environment for the bacteria to fix nitrogen. Because the environment will be essentially anaerobic and the plant will provide bacteria with ample carbon source. In exchange, the bacterium will send back to the plant this fixed nitrogen. So if you attended the previous webinars, specifically this one, the Genome Search Advanced Search Builder, then you know that the second example in this webinar showed you how to find isolate genomes of Bradyrhizobium from plant habitats, such as nodules, roots, leaves, and stems. Now let's build upon this example and let's extend it. The very first research question that we will ask is whether Bradyrhizobium is restricted to plants and soils or whether it is found in other environments as well. So the second question would be, if we find these Bradyrhizobium genomes in non-plant environments, are they capable of fixing nitrogen or not? And the third question is whether Bradyrhizobium from non-plant environments have nodulation genes or not. And the outline of the demo is that in order to answer the first question of whether Bradyrhizobium is restricted to plants or found in other environments, is we will do the 16S rRNA search of metagenomics database using a sequence from the type strain of Bradyrhizobium japonicum as a query, and we will use this tool called RNA homologs. And then we will use gene card and table configuration to see the environmental metadata of the metagenomes with hit. In order to answer the second question, whether Bradyrhizobium from non-plant environments can fix nitrogen or not, we will use another marker. In this case, we will use NIF-H protein, which is a typical marker of nitrogen fixation. Again, from the same type strain of Bradyrhizobium, and we will search IMG genomes to find genome-specific identity cutoff, followed by search of the metagenomes from non-plant environments to find the homologs of this gene. And we will use for this IMG top homologs and IMG genome blast tools. Lastly, we will take the nodulation proteins from the type strain as markers of nodulation and use the nucleotide sequences to search metagenomes with this Bradyrhizobium 16S. And for this, we will use workspace genome set blast. So that's the end of the introduction, and I will ask my colleagues now if there are any questions that I have to answer immediately.
Not really, Natalia. I think we can answer these by chat. Thank you.
Okay. So then we will proceed with live demo. So I am right now, so I am in my browser right now, and I am in IMG MER. And I can see it not only from this logo on top, but also it has my username, it has logout option, it has workspace, basically all of the features that I expect to see here. And again, here is the help button. Let's open it in a new tab. If you go there, there is this contact us link where you can submit a ticket if you have difficulty getting an IMG account. So how do I find, so let's start with the very first question of how do I find this sequence of 16S ribosomal RNA from Bradyrhizobium that I could use to search IMG metagenome data. So, and the answer is you have multiple options, of course. You can go to GenBank and find the sequence there, or you can go to one of the specialized databases, such as Silva, and find the sequence there. And also there are multiple IMG options, and some of them will be considered in the future webinars. But today we will do a very, very simple thing. So we will use the query that was used in the very first webinar about advanced genome search in order to find type strains of Bradyrhizobium. And to do this, I will click on this public list link, which will bring me to the set of public queries that were generated by IMG users and saved for other people to reuse. Specifically, I am interested in this third query, which is looking for genomes of Bradyrhizobium that have no more than 500 contacts, that are coming from isolate strains, meaning that they can be grown in pure culture, and they were isolated from an environment that is characterized by any of these keywords: nodule, rhizo, plant, stem, and so on and so on. I can select this query, and I can edit, click on Add Selected to History card. And this will populate my genome search history, so that now I will have this query that I can use and modify or just run directly. Again, if you are completely confused, I encourage you to go and watch the very first webinar about advanced genome search. So here is my query in the history, and I want to reconstruct it because I want to modify it somewhat. So after I clicked on this reconstruct, it opened this query in the advanced search builder. It shows me all of the query lines that I'm going to run. And as I said, I want to modify it because I want to search only for the genomes of the type strains. And because I am an expert IMG user, I know that I will find this information in cultivation metadata category in type strain field, and I will set it to yes. So if you are new to IMG, all of the fields and their categories are listed on the bottom of the page here, and you can use them in order to formulate your query. Now, if IMG can give me this query, which essentially lets me find the genomes that were isolated from the environment characterized by these keywords, why can't I do the same and look for the genomes that are not coming from this environment? And the answer is that absolutely you can do that. IMG has this capability, and I encourage you to try to run this query on your own. Two hints is that IMG allows you this not only AND operator, but also AND NOT, in which case it will be looking for the genomes that do not have any of these keywords in their environmental classification. So the second comment is that even though they won't have any of these keywords in their environmental classification, it doesn't mean that they will have any metadata at all. And that is actually the reason why we are running this roundabout way of trying to figure out whether there are any Bradyrhizobium strains from non-plant, non-soil environments. So now that I have kind of modified the query the way I want, I can click on search, and I can wait for the IMG to show me the result. And IMG gives you detailed messages about what exactly it is doing. That right now it is searching for the genomes of Bradyrhizobium in less than 500 scaffolds. It is looking for the isolate genomes from these environments. And the last thing that it will be looking for type strains. Now, why am I interested in type strains, and what is a type strain? Type strain is sort of an anchor strain that was used to characterize the species. It is the published strain, meaning that people have conducted a thorough experimental investigation, and they characterize it biochemically, phenotypically, and so on and so on. So basically, this is the anchor strain around which the rest of the strains are assembled that are representing the species. So IMG gives you detailed statistics about how many keywords, how many counts, and so on and so on. It was found in which field, and the final combination of this search is I have 8 genomes. So if I go click on the, and again, remember that this is to find the 16S sequence from the type strain of Bradyrhizobium. So if I click on any of these genome names, I will get to what is called genome details page. And in this genome details page, I will have annotation statistics, including links to all of the predicted genes in this genome, including 16S. So I will click on the name of this Bradyrhizobium japonicum USDA 110, which will bring me to the genome details page for this particular genome. Genome detail, any kind of details pages in IMG, they give you an overview of all of the data that IMG has about this object. And IMG has a lot of data about genomes, and that's why this page is so long. So it tells you when it was sequenced, how it was sequenced, who sequenced it. It also tells you about what is the taxonomy of this particular genome. It also tells you where it was isolated, where does it grow. And this genome, this particular strain was isolated from soybeans in '57. And on the bottom of the page, it gives you the statistics of genome annotation. It tells you how many genes in total are in this genome, how many of them are RNA genes, 85 in this case, how many of them are protein coding genes, and so on and so on. So all of these counts that you see in this table, they're linked to the lists of genes. And that is exactly what we are going to use in order to, the 16S gene, right? This is exactly what we are going to use for our query. I will open this, if I click on it, I expect to see a list of 16S genes, and I will open it in a new tab. So in this case, I will probably get a list of 1, right? Because there was one 16S gene in this genome. There is a list of 1. And it gives you again some details about where this gene is located. But what I'm interested in this case is I want to click on the ID of this gene, because it will bring me to the gene details page. So here is another example of so-called details pages in IMG, which gives you all of the information IMG has about this particular object. In this case, it tells you that it is 16S ribosomal RNA. It is found on this particular contig. Within these coordinates, the length is almost 1,500 nucleotides, and so on. Next, it shows you the chromosomal neighborhood of this gene. The gene that I'm looking at is always in red, and if I mouse over, I see its annotation. If I mouse over other genes, I see their annotations as well. So, for instance, next to it, there is 23 ribosomal RNA. But what I'm really interested in this page is this bottom section, which has this RNA homolog option. So what I will do in this case, I will go to this menu of selecting homolog type, and I will click on top metagenome 16S RNA hits. This will start a BLAST job. IMG will take the sequence, nucleotide sequence of this particular rRNA gene, and will start a BLAST against the database, which will include all metagenomic 16S rRNAs. It will take some time because there are a lot of metagenome 16S rRNAs, and while it is running, you may want to ask a question. What if the genome that I'm looking for is not an IMG? What if the sequence that I'm looking for is not an IMG? Is there any way for me still to run a search against IMG 16S, for instance, databases? And the answer is absolutely yes. These options are located in this menu, Find Genes, BLAST, RNA. Again, I will open it in a new tab. So that, because you see, it says, do not close this page or your results will be lost. So we will go to a new tab, to a new page. So this is what RNA BLAST menu looks like. So in this case, you are required to paste an input sequence here, the query sequence here, right? So this is the window where you would paste it. So the sequence has to be in the FASTA format. If you don't know what FASTA format is, we discussed it in the second webinar about data export and download. So it also has the options. The only option here is BLAST-N, because you can only compare DNA against DNA. But you have a choice of BLAST BLAST database, which includes these public isolates, 16S, and different types of RNAs from metagenomes. Then you have the e-value options and the number of hits. So the databases against which you can run BLAST are described here. It also has the timestamp of when this particular database was generated, because they are not updated daily. So the options include this RNA from all public isolates, the 16S from public isolates, as well as multiple options from metagenomes, 16S, 5S, 18S, 23S, 28S, and all other RNAs. So, and the databases are exactly what they say, right? These are public assembled metagenomes. They do not include any kind of amplicon data. They do not include any kind of unassembled reads and so on and so on. So in order to show up as an assembled 16S sequence in these metagenomes, the lineage has to be relatively abundant, because it has to be showing in the sequence, and then it has to be assembled, and so on and so on. So that is the disadvantage of this kind of databases, that it only samples the most abundant sequences in the community. On the other side, the advantage is that it is completely unbiased, in a sense that unlike amplicon data, which starts with primers and has PCR bias and so on and so on, this data is completely unbiased. What it doesn't say here about what is true for any kind of searches in this Find Genes BLAST is that you are not limited to one sequence here. You can submit multiple sequences as multi-FASTA, but the requirement is that they have to have no more than 10,000 characters. So, and while we were talking about this option, our BLAST results came back. So we will close this window and we will go to these BLAST results. So we started again, as a reminder, we started with the 16F query, rRNA query, which we ran against all metagenomic 16F rRNA, and we've got this, what is a typical IMG homolog table. It shows you gene ID of the homolog, it shows you what the product name of this homolog, in this case it is ribosomal RNA, and the percent identity between my query sequence and this subject sequence, which is very high, almost 100%. Then it shows you other parameters of this of the similarity, such as e-value, essentially 0, and very high bit score. And then it shows you the name of the metagenome from which in which this gene was found. And in this case, it is soil from Hawaii. And the second best hit is from Arabidopsis root communities. And then again, soil, this time from California, from Anza Borrego. So far, all of the top hits are from the environments where we expect to find Bradyrhizobium. Let's scroll a little bit further down. So here we have something unexpected. Here we have a cell enrichment from seawater from North Sea. And then the next sequence is from West Antarctic, from coastal water. So now these are the communities where we don't expect to find the Bradyrhizobium, because we assume that it was only soil, plants, and everything else. Now, is it still Bradyrhizobium? And the answer is yes, it is, because these are again nearly identical sequences, 16S sequences. So as an answer to the very first question of, does Bradyrhizobium live only in plants, or is it found in other environments? The answer is yes. It is found in other environments as well, specifically in these marine communities. Now, this is kind of, we sort of answered this question, but if we want to be more thorough, and if we really want to see what is the environmental distribution of Bradyrhizobium, we would not stop just at browsing, kind of scrolling through this list of metagenomes. We would actually try to see their environmental metadata. And the very first thing that, and in order to do that, we will select some or all of these genes, and we will add them to gene card, where we have an option of table configuration showing whether the what is the environment from which this metagenome was sampled. But first, in order to do this, first we have to figure out, do we have only Bradyrhizobium hits here in this table? Or do we have some other genera and maybe families, orders, and so on and so on? In order to do this, what we can do is we can sort this table by clicking on the column header. In this case, I would click on the header of the column percent identity. The first click will sort it in the descending order, so that I will see the highest hit first. But the second click will sort it in ascending order, so that I will see the lowest hit first. And even this lowest hit in this case is nearly 98% identical. So, and lucky for us, people have studied 16S long and hard, and including the fact that they have formulated these cutoffs for the different lineages. And the general agreement is that if you have identity, percent identity between 16S RNA sequences of more than 96%, you are likely looking for the sequences from the same genus, maybe even at the same species. So this percent identity that I see, the lowest percent identity, it tells me that all of the hits in this homologs table, they are all coming from Bradyrhizobium genes. So I can safely select all of them and add all of these genes to GeneCart. So right now, IMG is collecting all of the attributes of the genes that we have selected and adding them to the GeneCart. And while it is doing this, okay, no, it's still not done. So let's get, I have this GeneCart preloaded for me. So this is what it will look like. It will tell you that there are these genes. These are the product names of these genes, the genome IDs, and then the genome names. So it's still not done. Okay, it's done now. So, and as I said, I can see the summary. Instead of the specific names of these metagenomes, I can see the summary of their environmental metadata. And in order to do that, I will scroll down to the table configuration options here, where I will select all of these gold ecosystem fields. Gold ecosystem category, subtype, type, and so on and so on. And after that, I will have to click on display genes again in order to see these additional columns in the table. In this case, basically, the table includes these columns. And now let's kind of simplify this table so that we can see the fields that we are really interested in. So I can do it using this column selector option. Again, it was discussed in the previous webinars. I can hide all of this gene ID, gene product name, because these are not very interesting. I can hide the batch. And after I close this configuration table selector, column selector, I have this table, which has the genome name, the name of the sample, of the metagenomic sample, the gold ecosystem, ecosystem category, subtype, type, and so on and so on. So I can sort the table by this gold ecosystem category, which is a relatively high-level description of the environment from which this metagenome came from. And I can see that on top, I have the aquatic communities. And these aquatic communities are quite different. Some of them are marine, and some of them are freshwater, and some of them are even coming from hydrothermal springs. So after that, I have some communities that are coming from bioremediation bioreactors usually. So, and some of them are bioremediation thiocyanide, and then there is metal bioremediation from uranium contaminated environment, and so on and so on. So, and then we have the plant communities, and then we have the bulk soil coming from the soil itself and from the peat, and so on and so on. So, and that is the question basically. And as you, again, if you watch the webinars, you can export this table and you can see the summary of the data. How many sequences of Bradyrhizobium are coming from plants, how many of them are coming from terrestrial environment, aquatic, and so on and so on. And so that completes the answer to the very first research question. Where does Bradyrhizobium live? Is it found in plants and soils, or is it found in other environments as well? Are there any questions right now?
There are definitely lots of questions. So I'm going to cherry pick one that may be most relevant. And it's from Matt, who asks, you know, if you're looking at not a 16S sequence, but any other sequence, what is the correct threshold of percent identity to use to, you know, say that you have a good hit?
That is a great question, and we are going to address it basically when we will be looking at the second research question in this demo. So, anything else?
Yes, Natalia.
Okay.
People are asking how to diagnose blast against 16S, 18S, etc. I say that you will describe it later.
I'm sorry, I didn't quite get it.
Users are asking about blast against 16S or 18S or other non-coding RNA genes.
No, anyway, so basically, probably this is the question that will be answered in the transcript, because I'm not quite sure what is the specific question. The answer is yes, you can run blast, but anyway, so let's proceed with the second part of the demo. Let's proceed with the second question of the demo, which is, so we have all of these metagenomes from non-plant, non-soil environments. So are they still capable of fixing nitrogen, or are these Bradyrhizobium doing something completely different? So how can we answer this question? In order to answer this question, first we have to add these metagenomes from non-plant environments, such as, for instance, these aquatic metagenomes, to our genome card. And in order to do this, first of all, I will deselect all of the genes that I have in my gene card. And the second thing that I will do is I will filter this table by the field gold ecosystem category. And in this case, I want to see the aquatic environments only. And that's what I am typing here, aquatic. Next, I will click on apply. And the table right now has only the entries for the genes that are coming from the metagenomes from aquatic environments. Now I would want to select all of them, and I will want to add them to the genome card. And the reason why I want to do that is because my next analysis will be limited to the genomes from non-plant environments, specifically from this aquatic environment. So now that we have them selected safely in the genome card, now let's go back and let's try to figure out which gene can we use as a marker of nitrogen fixation. So, and if you are familiar with this kind of analysis, most people use, even for amplicon data, most people will use NIF-H gene. So how can we find this NIF-H sequence in the same type strain of Bradyrhizobium USDA 110 that we have opened here, the genome details picture which we have opened here? So we can find it by looking for protein coding genes with function prediction. Again, let's click on this count and let's open it in a new tab. While it is opening, I can close some of the tabs that I no longer need. So this is the typical list, again, of genes. In this case, these are protein coding genes that have functional prediction. And if you are trying to click, basically follow me in this case, I see 4,900 genes, and you, if you are new to IMG, you may see only 1,000. Now, this is because IMG applied the default limits on the number of genes, hits, and everything else. And you can change it by going to this My IMG Preferences. Again, let's open it in a new tab. And here in Preferences, as you can see, there are settings for limits for all kinds of objects you will see in IMG. And by default, IMG is quite stingy. So by default, it will limit you to 1,000 genes. But there is nothing restricting you from changing it upward. And the same thing for maximum homolog result lists. You can, again, also change it to whatever number you want. But then you have to click on Save Preferences, or else it won't take action. So that's how you can see the full list of genes when you click on this gene count. So in order to find NIF-H gene, I will do the same gene table filtering that I showed you in gene card. So in this case, I will filter this column by gene product name, and I will type NIF-H. And I would hope that it will be found in this genome. So let's click on Apply and see what happens. So the table was filtered, and now I have only this NIF-H protein showing up. And it is an iron protein subunit of nitrogenase. Now, in order to get to the sequence of this gene, again, I would click on gene ID of this gene, which will bring me to the gene details page, which will have both nucleotide and protein sequences in this case. And also, it will have the same BLAST options, where IMG will go grab the sequence of the protein and put it in as a query for the BLAST job and so on and so on. So this page, this gene details page is much longer because IMG knows a lot more about this protein than about 16S sequence, including its assignment to protein families, metabolic pathways, and so on. So again, the same story about the chromosomal neighborhood, including the gene that I am looking at and the neighboring genes. And on the bottom of the page, we have these options for sequence similarity search. So, and now we are going to the question that was asked in the interim. The question is, what would be the appropriate BLAST percent identity cutoff for this particular protein when we are running BLAST against metagenome databases, so that we know that the hits that we are looking at are coming from Bradyrhizobium and not from some other lineage. Unfortunately, in this case, people didn't bother for us to run extensive research and formulate this percent identity cutoff. But then IMG is your friend, and IMG lets you do it on your own. So in this case, how we can establish this BLAST percent identity cutoff for the lineage specific, for the NIF-H from Bradyrhizobium and not from some other lineage? Well, we will run LAST against all IMG isolate genomes, and then we will find the lowest cutoff, the lowest percent identity of a hit from Bradyrhizobium, and that would be sort of the lowest boundary. So if the gene is below this percent identity, it's unlikely to be from Bradyrhizobium. Let's do this. Let's start this top homolog hits. And it may take some time because I think that a lot of people are clicking right now on different pages on IMG. So I have it preloaded here. I have this page preloaded here. So after this search has run, after that you will see again the same type of page, the homolog display page with the gene ID of the homolog, the product name of the homolog, percent identity, alignment on the query and the subject, length, other parameters like e-value and bit score, the name of the genome, and so on. So now we can filter this homolog page, this homolog table, by selecting only the genomes from Bradyrhizobium. And I will select this genome name column, and I have no idea why it doesn't want to do it. Yeah, genome name. And then I will type here Bradyrhizobium. I will click on apply. And this will filter the table, leaving only the rows that have Bradyrhizobium in the genome name. And as you can see, it is less than 200 hits. Now, in order to find the hit with the lowest percent identity to Bradyrhizobium, I will again sort this table by clicking on percent identity. First, descending filter, descending sort, and the second would be ascending, so that I will see the lowest percent identity. And the lowest percent identity of my query sequence between my query sequence and any other hit from Bradyrhizobium is 92%, right? So if I run BLAST against metagenomes and I find only hits at 80% identity, I can be confident that none of them is coming from Bradyrhizobium. Now there is the second question. So this is the lower boundary. But what if there are some hits from non-Bradyrhizobium genomes that have higher percent identity than 92? Can we find them? The answer is absolutely yes. And again, in this case, we would use the IMG table filtering options, except that in this case, instead of doing the simple text filtering, I will do this regular expression filtering. And I will type something that probably make no sense to any people who did not attend the data export and download seminar. and that's why we encourage you to go and watch it. But essentially, I am filtering the table by leaving only the rows that do not have a B in the beginning of the genome name. So if I click on apply, you see all of the Bradyrhizobium disappeared, and now I have some other genomes in this table. In this case, I am seeing again the lowest percent identity, and that's not what I am looking for. Actually, I am looking for the highest percent identity in non-Bradyrhizobium genomes. What I'm seeing right now here, it is a single amplified genome. Probably you don't see it if you are. Probably what you are seeing is the Rhizobium, this Rhizobium genome. And as you can see, its percent identity to my query is actually higher than 92. It is almost 94% identity. So what does it tell me? It tells me that if I get metagenome hits between 100 and 94% identity, I can be fairly confident that these are Bradyrhizobium hits. If I am getting hits below 92% identity, I can be fairly confident that I am getting some other hits from non-Bradyrhizobium lineages. And in between this 92 and 94% identity, there is this gray zone. I'm not quite sure whether it is the Bradyrhizobium or not. And I will have to use some other filtering and ideas about finding out what these are. So now that we have established this percent identity cutoff for the hits from Bradyrhizobium, for the NIF-H genes from Bradyrhizobium, now we can go and we can run BLAST against these selected metagenomes that we have. I will click again on this query gene because it will give me the gene details page again. I will go again to the bottom of this page. But in this case, instead of running this top homolog hit, I will use another option here. It is called IMG Genome BLAST. And this one allows you to run BLAST against your selection of genomes. As you can see, it can be no more than 100 of genomes or metagenomes, and by default, these are the genomes that are in my genome card. And that's why I was selecting the metagenomes from aquatic environment and adding them to my genome card. So again, it allows you to configure some configuration. For instance, it lets you find lets you set how many hits you want. And in this case, 10 will be fine enough for me. I'm not looking for all of them. And I can change also, decrease the E-value of the hit because I'm looking for nearly identical sequences. So if I click Run BLAST, in this case, it will run the BLAST job for me against these selected metagenomes. While it is doing that, you may ask a question. Can I do the same, but using my own sequence, which is not an IMG? And the answer again is absolutely yes. Again, the option is here in the Find Genomes, selected Find Genes, BLAST, selected genomes. If we open it in a new window, in a new tab, we can discuss what it allows you to do. Again, in this case, you have to provide the query sequence. Again, it is not limited to a single sequence. It can be multiple sequences in FASTA format as long as it is less than 10,000 characters. In this case, because you are running BLAST against genomes or metagenomes, you have option of BLAST-P or BLAST-X or BLAST-N, comparing protein versus protein, DNA versus protein, or DNA versus DNA. Again, you have E-value configuration options, and you have the number of hits, which you can set. It is limited, as you can see, to 100 genomes or metagenomes. And if it keeps basically, and we will discuss what you can use in order to increase the number of hits that you want to see. In this case, the sequence, the input sequence you have to provide yourself. IMG won't be able to find it for you. So this page is still loading, right? So basically, it is still looking for homologs. Now that it is finished, it found them. So here are the hits from the metagenomes. And as you can see, the very first top hit has more than 94% identity, meaning that it is likely the hit from Bradyrhizobium lineage and not from some other lineage. There are some other hits that have more than 94% identity. In order to figure out how many and what they are, we can again filter the table by saying that we want only the hits that have more or equal to 94% identity. I'm filtering the column percent identity with this number. If I click on apply, then I have only these hits that have more than 94% identity. And here is the why we went through the introduction about the percent identity versus the bit score versus the e-value. Here are the hits that have high percent identity, meaning that they are more closely related to my query sequence, but they have lower bit score because they are fragments. The full length gene is almost 300 amino acids, and these are less, about half of that. But still, these are the same marine environments, marine, seawater, and so on and so on, the peatland, and here are the hits to this nitrogenase protein. So this is the question, the answer to the second question that we have in our research topics, right? So do the Bradyrhizobium strains from non-plant, non-soil environment, do they have nitrogen fixation genes? Are they capable of nitrogen fixation? And the answer to this question is yes. They can, there are Bradyrhizobium strains in these environments that can do nitrogen fixation. And I will stop here and ask if there are any other questions.
Yeah, Natalia, I think this would be worth addressing from, yeah, it says, it's basically asking, we have this percent identity that we can select, but what about small, when percent identity is high, but small coverage, there is possibility of only conserved shared domain. It's filtering with both percent identity and coverage, which I believe is alignment length possible.
Yes, absolutely. That is an excellent question. Absolutely, that is, there is no kind of hard and fast cutoff saying that if you see this percent identity over this length, then you are seeing the hit from this particular lineage. So unfortunately, it is up to you. You have to use your judgment. So that's why I was looking, but kind of as a general comment, if I'm looking at a 300 amino acid protein and I'm finding hits of 160 amino acids, it is unlikely that I'm looking at just one conserved domain, right? So this is more reflective of the overall sequence conservation. If I was seeing hits of 50 amino acids and smaller, then you're absolutely right. I may be looking only at the hits to the most conserved domain of the protein. So that's kind of the answer is as good as I can give it. Any other questions?
No, thank you.
Okay, excellent. So then we will proceed with the third exercise of this demo. We will be looking for the so we established that there are Bradyrhizobium in non-plant environments. We established that they can do nitrogen fixation. So the last question is, maybe they even have nodulation genes, despite living in the environment where they have no chances of ever meeting a plant host. There is such a possibility, right? So how do we go around it? Well, again, in this case, we have to find these marker proteins of nodulation. And again, we are doing exactly the same trick. We are going first to gene details page. We are looking for protein coding genes with functional prediction. Here is the list of these genes with functional prediction. And again, we are doing the filtering of this table by the gene product name. And in this case, we will be looking for anything that is called nodulation. If I click on apply, so here is my list of proteins in this genome that have annotations that have nodulation keyword in this in their names. So and completely arbitrarily, without doing anything kind of sophisticated and so on, I will select these 4 proteins and consider them the signature of the nodulation genes in Bradyrhizobium. So in this case, I can either go to the gene details page of these genes, or I can directly click on Add Selected to Gene Card. And after some thinking and after some loading, now I have these genes in my genome card, except that I don't see them. So what happens in this case is that IMG, if you are adding one set of genes, another set of genes to your gene card, IMG assigns batch number to them. And so the latest batch will be the one that was, in order to find them, you would have to resort the table by this batch number. So now that I have the table sorted in descending batch number, so these are my 4 nodulation genes added to the gene card as the batch 3. So now let's clear all of them. So now what I want from these genes is I want nucleotide sequences. So again, if you attended the download and export webinar, you would know how to do it. Let's show it again. I will clear all genes and select only those nodulation proteins. And then I will click on this Upload and Export and Save tab, where I have an option of displaying the sequence of these genes, either amino acid sequence or nucleotide sequence. So in this case, I will select nucleotide sequence and ask to see them in this export format. So I have these genes, so I can basically copy-paste the sequences. So now let's copy them. So I have the gene sequences, my query sequences that I want to run BLAST against. So the second question is, what do I want to run it against? I can run BLAST against this selected set of metagenomes that I have in my genome card from the aquatic environment. But let's say that in this case, I want actually, I want not only these metagenomes, but I want all of the metagenomes, including those from plants, from soils, and so on and so on. So let's select all genes. And let's add all of the genomes to the genome card in this case. So now I have 196 genomes in my genome card, and that represents a problem. If you remember, I showed you BLAST, find genes, selected genomes, and that had a limit of 100 genomes or metagenomes. So what do I do in this case? In this case, I can create a set of genomes by selecting all of them, going to Upload, Export, and Save tab, and on the bottom, saving the genomes to my workspace. I will have to provide a name of this set, right? Or I can replace one of my existing sets, and I can do whatever name I want in this case, as long as it is non-redundant. For instance, I would want to save them to the set called Brady. So, and after I have done that, and because this is the job that I'm going to do is the request for computation job, I have already done this exercise and I have this genome set in my workspace. Again, you have this option only if you are in IMG AMR with login account options and so on. In my workspace, I have this set of metagenomes that I called metagenomes with Bradyrhizobium. And if we click on it, if I click on this count, I will see what these are, and these are exactly the metagenomes that I retrieved by this 16S search with Bradyrhizobium sequence. So I can select this set, and I can go to this BLAST tab in workspace, in genome workspace, where I can now paste my sequences, my query sequences, all of them, the 4 genes that I had. So I have to change it from BLAST-P to BLAST-N because it is DNA versus DNA comparison. And probably I don't need these 500 hits. I can do with 10 hits per metagenome. And then I can provide a name and submit it as a computation request. You see, in this case, it is not 100 genomes or metagenomes. It is now up to 500. So it is much larger. Because it is a much larger set, it has no chances of finishing in real time. And that's why you have to submit it as a request for computation. You have to provide the job name. In this case, I submitted as a name, Brady Blast, or whatever other name. After you click on Submit for computation, you will get a message that your computation has been submitted, and you will be notified by email when your job is complete. So if you have very stringent spam filtering in your workspace, you can still, in your email account, you can still find your completed jobs if you go to this workspace, My Jobs option. So in this case, my job was Brady Blast, right? And it was looking for genome set metagenomes with Bradyrhizobium, and it was blast 10, and it was e-value of this, and so on and so on. So if you click on this completed, and when it is completed, when the status is completed, you can click on it, and you will get the results of this particular search. As you can see, it was nucleotide search of these nodulation sequences, and it was against the database of 195 metagenomes. There is an option of looking at the raw blast data file. And here is again the same homolog table that we have seen in IMG again and again. So what we are interested in is the list of metagenomes in which we found hits to these nodulation proteins. And as you can see, just from kind of eyeballing it, it is all root nodule metagenomes, root nodule metagenomes. So far, it doesn't look very promising, right? It looks like the only environment where we find the nodulation genes seem to be the root nodules and nothing else. Again, you can continue with the exercise of scrolling the table, or we can again use the IMG table filtering option. Let's say that we no longer that we don't want anything that has the root in the name. So again, we will set it to regular expression, and we will filter for anything that doesn't have that doesn't start the names that do not start with R. In order to figure out what I am doing, again, you are encouraged to watch the download and export webinar. So now our filter excluded any metagenome names that start with R. What we have is we have this peat permafrost and another permafrost soil. Notice that we do not have any marine communities, no seawater, no groundwater, no bioremediation, no nothing, right? So basically, so it looks like in the aquatic environment, Bradyrhizobium strains have nitrogen fixation, but no nodulation genes. Now, what about these peat and permafrost soils? As you can see, percent identity is quite low. It is only 77%. Are there still hits coming from Bradyrhizobium or not? So in order to figure it out, you can look at the scaffold on which we found the hit. Again, we will open it in a new tab to look at it. By default, you are redirected to scaffold details page. Again, details, the same details that you will see all of the information that IMG has about this particular scaffold, including the predicted lineage. When IMG annotates metagenomes, it tries to assign the taxonomy to each and every contact and scaffold. And this is what it tried in this case. And what it assigned in this case is it is not Bradyrhizobium. It is actually a Burkhaldium, which is a completely different lineage from a completely different even phylum. No, from a completely different class. No. So as you can see, basically, so what we can say basically based on these results is that, and if we remove this filtering, and we will look back at the table that we have of homologs that we have, and if we review the percent identity, blast percent identity, is that most of them are above 90% until we get to these peat communities where it suddenly drops from essentially 90% to 70%. So, and it looks like the only Bradyrhizobia, when it is no longer a hit to Bradyrhizobium, but instead of to Burkhalderia. So this is what it tells us, that essentially the Bradyrhizobium that have nodulation genes are found exclusively basically in these legume communities, in the root nodule communities. While the Bradyrhizobium it is a very ubiquitous microbe, which is found in many other environments, except that they no longer carry nodulation genes there. And that concludes the second part or the third part of the presentation. Any questions?
Natalia, I think since we're at 12:12, I think it's worth saying that we will work on answering these questions that we perhaps did not get to during the chat. So we will either provide better answers or get to your questions and email a copy of the chat transcript.
Okay, so thank you for attending again. We apologize for running over time. We hope that it was informative. We will post the recording probably by the end of this week, together with the chat transcript. If your questions weren't answered live, we will add the answer to the transcript of the chat, and we will post it together with the recording. You will get an email notifying you of the posting of the recording, and you are most welcome to review it again. If you have questions about using IMG and so on, so you are again encouraged to use this contact us link in the help section of IMG. Thank you so much, and I hope